Hello everyone, my name is Jerry James and this presentation is based on the paper Diamond-Based Nanomedicine, an enhanced drug delivery and imaging, which was written by Moore et al. And published in 2012. I'll be focusing on one key aspect of this paper, the use of nanodiamonds for imaging, and I will explain how nanodiamonds work as imaging tools and what makes them uniquely valuable compared to traditional materials like dyes or quantum dots. Nanodiamonds are incredibly tiny carbon particles. They are just 4 to 6 nanometers wide. That's thousands of times smaller than a human hair. They are not mined like jewelry diamonds but produced synthetically using techniques like detonation or laser ablation. They keep the strong, stable crystal structure of diamond, but at the nanoscale, what's fascinating is that they are not toxic and are biocompatible, which means they are safe to use in living organisms. They also tend to group together into clusters about 100 to 200 nanometers wide, which helps them to stay longer in the bloodstream. What makes nanodiamonds so powerful for imaging is that they can fluorescence. They glow under certain lights. This is due to a tiny structural defect called a nitrogen vacancy center or NV center. Basically, a nitrogen atom takes the place of a carbon atom, and next to it is an empty space, a vacancy. This defect causes the nanodiamonds to emit stable red light when hit with a laser. Unlike traditional dyes, nanodiamonds don't photo bleach and they don't fade and they don't blink. That means they can stay visible for hours or even days inside cells. Fluorescent nanodiamonds can be used in a number of imaging applications. One is live cell imaging, where scientists observe how cells move and function in real time. They are also used in intracellular tracking, helping researchers watch how molecules or drugs travel inside the cells. In MRI, nanodiamonds can even enhance contrast when coated with gadolinium, and importantly, their surface can be modified. For example, to carry antibodies or ligands, which lets them target specific cells like cancer cells, which was a major invention by the humankind for the medical needs. Now, advantages. Let's compare nanodiamonds to traditional dyes or quantum dots. Dyes often fade quickly. This process is called photobleaching. Quantum dots are brighter, but they contain metals that can be toxic to cells. Whereas nanodiamonds solve both the problems. Their light is stable and long lasting, plus, they are made of carbon, a material that's naturally compatible with the body. And because their surface can be customized, they can be attached to targeting molecules or for personalized imaging. This includes detection of cancer cells. When we look into the highlights of this paper, the paper gives solid evidence of safety and effectiveness. In one study, nanodiamonds were injected into mouse lungs and showed no signs of toxicity. In another, they were introduced into the reproductive system of worms and were passed safely to the offsprings with no harm. They were also used to target cancer cells by attaching folate, a vitamin that cancer cells absorbs more. This lets researchers visualize the cancer cells clearly under the microscope. Of course, there are some challenges. We still don't fully understand how nanodiamonds are cleared from the human body, and scaling up production to clinical levels is still being worked on. However, researchers are making progress and the potential is huge. Nanodiamonds could soon become a tool for personalized medicine allowing real time, targeted, and safe imaging for individual patients. In summary, nanodiamonds are one of the most promising imaging tools in nanomedicine. They are safe, stable, and precise. With continued development, they could change the way we track diseases, visualize cells, and personalize treatments. Thank you for listening.